Computer. All right, let's go ahead and get started if that is okay with everyone. I guess even if it's not okay, because it's okay with me. So we are going to dive in today. I'm really excited. We're going to talk about, well, last week we talked about Venom. We're going to do a quick review of some important points. Last class. Today we're going to actually talk specifically about evolution of Venom. And we're going to talk about it at a few different scales. But before we get too far into that, couple things we're going to do. The first thing, just want to show you the agenda for today. So I'm going to, right after this, want to cover briefly the Herpetology in the News article that I sent to you guys. And then we're going to give you some work time to do the heroes and villains activity that I assigned last class. And then after that is we'll have this big evolution lecture, hopefully not too big, hopefully more interesting. And then at the end, we've got a debate that we're going to talk about. Now, I don't want to give any hints about what that's about at the moment. If anyone's really familiar with Venom and reptiles, you might already have a hunch. We'll see. Any burning questions before we get started? All right, great. So first thing is, oops, that's the wrong slide. Where is my Herpetology in the News? So this is an article, just show of hands, who looked or thought about, who thought about looking at the article? Okay. So no one looked at the article. That's okay. So hopefully you guys can go to the program from the other day and look at this article that was Herpetology in the News. So this was a study about venom in diets that came out last year, I think. Yep. And the article that I sent to you guys is a summary that does an interview with a couple of the researchers, the researcher who led the work and the professor whose lab a lot of the work was done in. So the idea behind this study was they collected genetic data from all of basically the rattlesnakes and the moccasins, so the Echistrodon. We actually talked about these two genera last week, Crotalus, Echistrodon, and the pygmy rattlesnakes, Cistrus. So they have this great big phylogeny. They've got venom samples from all of these snakes. And we have a special type of data that's called the transcriptome, and it's specific to the venom. But basically, all that it allowed them to do is determine roughly how complex these snakes' venoms were. And then, so they have these data, and they compared it to data from their diets that was collected from many, many natural history notes. And with those, what they were interested in was evaluating some different hypotheses for the relationship, if there was one, between the complexity of venom, which means like how many different components you have in your venom and maybe how evenly expressed they are, and the complexity of the snake, of the snake's diets. And what they found is that it's actually not about diet per se. It's not about how many species you're eating. What's really important for these snakes is the phylogenetic diversity of the things you're eating. And so I'll give you an example because you might be thinking, well, what does that mean? Think about it like this. If a snake is eating 10 species of mice, it's 10 species, it's a lot, but they're all kind of mice. Not that big a deal. Versus if a snake is eating one mouse, one lizard, and one bird all the time, that is a much more phylogenetically complex diet. And it turns out that if you're doing that, if you're a very generalist feeder, you have to have a more complex venom. So a couple of the things that this article showed, it showed this idea that complexity is correlated with the phylogenetic diversity of prey. That's really important. And this ultimately provides support for the idea that selection is shaping venom in these species. And a really sort of great metaphor. This was a quote by Dr. Matthew Holding, and he said, you can think of venom as the snake's toolbox. And if you have a toolbox with a wrench, a socket, screwdriver, all of those things has a different use, and you can envision a scenario where they might be useful. Similarly, each protein in the venom has a different function. And when injected into a mouse, lizard, centipede, that's going to provide a different function and maybe make it better or easier to eat. So Any questions about that? I know that's a quick summary. If you guys have any questions about that after you've taken a look at that article, because remember, we can have quiz questions come directly from the herpetology in the news things. Please let me know. With that, I think this is a good time as any to break into your groups that you found on Monday. Yeah, and start working on the superheroes and villains activity. We want to give you guys about 20, maybe 25 minutes to work on that before we dive back into some more lecture. Okay, let's go ahead and start bringing it back for our little lecture portion. So as people are moving back to their seats and everything, I'm going to start. Let's chit chat about some of the stuff that we covered last week or on Monday. I shouldn't say last week because it's just Monday. So we talked about venom. We gave ourselves a definition for it. We said it's a traumatically introduced natural toxin. Remember, that was one of those things I said, be sure everyone knows we have our definition for venom. And then we also talked about what venom actually was in this very like biological sense, this mix of proteins that have different functions, things like that. And we also talked about some examples of venom variation. So I want to talk about, remember these guys, these are palm pit vipers that are very similar. They're two different species, but they're sister taxa. And even though they live in similar places and things like that, they have these very, very different venoms. We remember this guy has this neurotoxic venom. This guy has a hemotoxic venom. That means one attacks your nervous system. The other is attacking your blood and tissue. And we talked about another species that shows that same dichotomy that occurs within species. These are Mojave rattlesnakes. And then we closed out our discussion talking about some of the diversity of venomous reptile and in a couple cases, amphibian lineages. And so it's here I want to actually pick up today. And the first thing I want to mention is, we talked about these groups, and I think I might have used these terms, but I don't know if I classified them like this. But one way to differentiate these general lineages is by high pressure and low pressure. So in the top, we've got our what we think of as our like venomous families, the ones that we've known for a long time. We have our Elapidae on the far left, our cobras, kraits, coral snakes. We have Viperidae in the middle. These are going to be our general vipers, as well as rattlesnakes, moccasins in the US, tree vipers, lance heads. And then on the far right is the Atractaspidae. That's that group I mentioned. It's kind of funny. The stiletto snakes, they have a really long set of fangs. We're not going to talk about them as much today. And then contrasting that, we have our Colubridae on the left. Those are our rear fang snakes, and Helodermatidae. So what is different between these two groups is these guys have these very well-developed fangs. They're often, or they're basically always at the front of the mouth where they can, be used to bite things, or in the case of Atractaspidae, stab things. They have a very developed venom gland system. And attached to that gland is a series of muscles that when contracted will force venom out. So you can think about it like if you had, for instance, a water balloon, and maybe you have a straw in it. If you squeeze that with your hand, you're going to shoot a jet of water out of it. That's what these guys do, basically. In contrast, these low pressure systems, they don't have these complete fangs. Their fangs tend to be grooved as opposed to like a needle. And their gland does not have that same kind of muscle attachment that goes around it. So instead, they have to create pressure by biting and move some of their venom with capillary action. You might think about this. I was thinking about these glands, how would you describe that? It's kind of more like you have a sponge. You kind of squeeze the sponge and it like drips out, and ideally you would catch it. Here they catch it with the fangs and it gets into the prey. Yes, Ethan.
So does that mean that in the low pressure systems that the venom is constantly circulating in their mouth as a part of their saliva?
Nope, not at all. So think about it like if you, for instance, actually your own salivation. So most of the time we don't go around salivating like crazy, but when you get hungry, or there is an external cue, you will dramatically increase your salivation. And so it's the same with these guys. They're not like constantly, they're not constantly producing this, but once they start chewing, it will like kick everything in and then they'll start producing the venom.
Does that mean they've evolved their own immunity to their venom? And like in case of things like mouth sores and things like that, and not getting into it?
So this is something I'm not going to talk about very much at all, in fact, but actually almost every species has some degree of immunity to its own venom. And these occur with what are called small molecules called inhibitors, that you can find them in the bloodstream, for instance, and things like that. But I'm not going to talk about that. And then the other thing to remember is, of course, that they have their own, have a degree of like control over when they're actually, letting their venom out. But so we have all this diversity, and especially one of the things that I made a point to mention last lecture was that we have a lot of diversity in the fang structure. So our vipers have those selenoglyphous fangs, our elapids have proteroglyphous fangs, and our colubrids have epistoglyphous fangs. This is really interesting, but there is this question of where do all these fang morphologies come from? You might also think about this as how did these fangs evolve? I think this is a really interesting question because one of the things that's really neat and probably maybe not intuitive if you're just looking at skulls, but in fact, all of the fangs of our fanged snakes are all attached to the same jawbone. So even though their heads are very different, the structure that their fang is attached to is homologous. So up here, we have on the far left a phylogeny that shows the evolutionary relationships of these species and some representative skulls that are just images. And on the right is what I really want to focus on here. So these are cartoon representations of the skull. And what is highlighted in red are homologous structures. So these are parts of the skull that are evolutionarily from the same ancestor. And you'll notice that in all of these cases, that is where the fang is attaching. So for instance, we have our rear fang snakes and they have their fangs far back in the mouth. Well, it turns out that really vipers and elapid snakes have their fangs in basically the same place. It's just that bone has been migrated forward. And so in fact, this emergence of this trait of being front fanged that we see in vipers and elapids, it evolved convergently from epistoglyph ancestors. And that means that, ancestrally, they all probably had these sort of like rear fangs, and then they now have evolved the front fangs. And so one question we might ask ourselves is, how do we really know for sure that they came from an epistoglyph ancestor? It's not likely, but it is hypothetically possible that these are three convergent emergences of fangs, or maybe there was two independent emergence of front fangs from the aglyphous or non-fanged ancestor. Well, actually, we have evidence from embryonic studies or developmental studies. And if you highlight the tissues that produce these bones, it's kind of funny, actually, as, for instance, vipers develop, you can see the tissues migrate from the back of the head to the front of the mouth as they take their final skull shape. Yes, Sydney? Yeah, sure can. Yeah. Yes, Princeton.
I guess, sorry,
You're good.
Going back to the venom delivery systems.
Yep.
I guess my question is, would there be like selection on that low pressure system to have increased, toxicity due to the fact that there's less venom?
It could be. I would say, so it's kind of a tricky question because especially in colubrid snakes, there tends to be a lot of variation in just general life history. So one of the things we know about, for instance, vipers and elapids is that by and large, you need to have venom. And if you don't, they're so reliant on this trait that it's just a critical part of their life history. With colubrids, there are lots of cases where it seems like venom might be helping, but maybe isn't a critical component. So for instance, maybe you're like a cat-eyed snake that's preying on birds. And even if you didn't have venom, you could still catch and constrict birds. And they often exhibit like constriction-like behavior. but maybe these toxins are helping speed up the process. So it's hard to say, and selection can be challenging that way because it's a mix. It's going to be a function of all those ecological forces, but certainly there are cases where that could be. So yes, Ethan.
Do we see in like the venom gland themselves? I know some are like at the top, some are at the base.
Yeah.
Is it the same sort of like basal structure? And both, like, are they both like some sort of modified salivatory gland?
Yes. Yeah, they're basically always a modified salivary gland. And in vipers and elapids, it's actually very clear that they are from this ancestral, gland state of that's like characteristic of the epistoglyph snakes. With the epistoglyphs, it's a little, or with, I should say with Colubridae more broadly, there's actually quite a bit of variation even in the gland and fang structure, and far more variation than almost anything we see in vipers and elapids. And so that is a little harder to track down exactly what, the evolutionary histories are. Okay. Sydney, are you good on this guy? Or you need a couple seconds? Okay. So while Sydney is finishing up, I'm going to prime us for the next one. So one of the things we talked about, I want to mention this as a reminder, we talked a little bit about some different life history strategies between the Viperidae and the Elapidae. So for instance, we mentioned these differences in things like sit and wait versus active foraging, what kind of effects those might have. And so with that, we've been talking about a lot of diversity. I want to pose a question. It's going to be a two-parter, and I hope the answer to the first part is no. Does anyone know what this snake is? You do, Ethan. Then I don't want you to say what it is. So everyone who does not know what this snake is, can I get a show of hands for who thinks that this snake is a viper? So family Viperidae. Yeah. Okay, so here are your options. Family Viperidae, family Elapidae, family Colubridae, because it has to be in one of those three families. So show of hands for family Viperidae. I got, you can just raise, okay, couple. Family Elapidae, one. Family Colubridae. So I got a lot of people not raising hands. That's okay. If you want to hedge your bets, that's all right. So this snake, Ethan, what is it?
Is it a death adder?
It is a death adder. So this is in fact an elapid snake. Now what's cool about death adders, at least I think it's cool, is that they have a lot of morphological similarity to our vipers. And death adders are only found in Australia. And another unique thing about Australia is that there actually are only elapid snakes in Australia. There does not seem to have been a migration or an invasion of viperid snakes into Australia. And so it is hypothesized that the kind of strange from an elapid standpoint, this is kind of a strange looking snake, but it's hypothesized that it's because these guys are filling a viperid or a viper-like niche. And so they have evolved this convergent morphology that includes things like a heavy body, a very fast strike speed. They're definitely a sit and wait predator, which is just kind of a neat thing. And so since we're talking about convergence, I want to talk about one other really cool, really neat, and perhaps surprising example of convergence. Sydney, you good on slides? Nope, a couple more minutes, that's okay. Yep.
In convergent ones like that,
Mhm.
do we see more of a, well, actually, never mind, because I guess you said they were high pressure, so
Yeah.
that would be the same.
Yeah, absolutely. All right, so another example of convergence, spitting. So when I think of spitting cobras, I think of just a spitting cobra. I guess I know hypothetically that there are spitting cobras in Africa. I know that there are spitting cobras in Asia. You may or may not know those things, and that's a-okay. But as it turns out, this emergence of spitting is in fact convergent, and it is convergent 3 times. So one of the more obvious examples where you can tell it's convergent on the left is a group of elapid snakes called the rincals, and on the right is a traditional spitting cobra, just an African spitting cobra. So look at the difference in how they spit in this sort of behavioral spitting. The rincals have this very obvious where they kind of throw their body forward, and that's how they, get their venom out. Whereas these other African spitting cobras are just like really fast, just kind of tilts its head up, venom comes right out. And those are due to some small differences in Fang morphology, but those are indications that this is convergent evolution. And in fact, if you look at the full phylogeny of the rincals and cobras, it turns out that even among spitting cobras, for instance, in Africa and Asia, spitting has emerged independently. So this is a phylogeny. I know it is kind of funny looking, and just bear with me. Over here in this section of the phylogeny, these are all of our other elapids, not the things of interest. So our first lineage that we're curious about is the rincals. It's right here. You can see that it's marked with this, this indication that this is a spitting lineage. And then over here, we have a spitting lineage that's sister to a non-spitting lineage. We have a spitting lineage that is sister to a non-spitting lineage. And so those are indications that these are in fact convergent events. Now, what's really cool, or one of the things that's really interesting about this, is that in these cases, we have this very clear morphological and behavioral convergence. We actually also see convergence in the venom phenotypes as well. So all of these groups that have the spitting, they actually have an increase in this group of toxins called PLA2s. And in the paper that showed this, which came out only last year, they basically show that the combination of this toxin, these PLA2s, with another group increases the amount of cellular activity and basically the amount of pain that you're causing. And so it's basically a synergistic interaction of the venom to increase pain. So you're spitting at an organism, ideally you're aiming for something like its eyes, and you want an immediate response, you want a pain response, and that's what they've convergently evolved to do. Yes, Sydney.
I was just going to ask, sorry about the dark powder.
Mhm.
Are they, because I know with looking at it, it has a big head,
Mhm.
and it looks like a viper, so are they going to be like more morphologically similar to vipers?
Well, I mean, their body structure is, right? But actually their fang morphology is not. So their fangs are still proteroglyph fangs, they're short fixed fangs. I don't actually know as much about their venom glands. Presumably they're a little larger than most elapids because they have that larger head, but I don't actually know that. So actually in some ways, one of the most interesting things to me is why is their head kind of triangular like the vipers? And again, I would presume it has to do with their gland structure, but I don't know that. Okay. So this is a really cool thing. And I wanted to present this here because I want it to lead into what we're going to talk about next, which is the evolution of venom itself, of this variation that occurs in venom. And so to get into that, I first actually want to go back to basics a little bit. So this is sort of early biology, bio 1, bio 2. Central dogma. Do you guys remember this? Yeah. So this is the idea we have DNA, RNA, protein. That's the way things go. We have genes that are coded in our genome. Many of those genes produce a protein product, and there's this intermediate step that's the RNA. Now, what's important here, your protein is ultimately going to be what determines your phenotype. And this is a general statement in biology. Most phenotypes are a function of their proteins in some way, shape, or form. Now, what's really useful and powerful about venoms is that because venoms are ultimately just a mix of proteins, we have a very clear path to look at this, right? So you might contrast thinking about the evolution of venom with the evolution of another trait. For instance, arms. Think about how many genes underlie the evolution of an arm, and how many genes interact in the evolution of an arm. Those sorts of things are really challenging and difficult to tease apart. But in venoms, it's basically what proteins do you have and how much of them are there? And we can identify where those changes occur at each of these different steps. So for instance, we can say, what genes do we see that underlie the venom? What are the sequences of those genes? Because we know sequence is going to affect our protein structure and function. And then we can also look at gene regulation, which is how much is that gene expressed? Is it upregulated? Is it expressed a lot or not? And remember, we know that gene expression is something that tends to be tissue and time specific. So here we're talking about expression in the gland, and that's actually a very important axis of variation. So speaking specifically about venom, we would say that our venom composition is shaped by two characteristics: the available toxins and their expression. And if we look at a plot that looks like this, our bar chart, that's actually what we're looking at. Here we're looking at what toxins are available in the genome and how much they are expressed, how high each of these bars are. And these two features correspond to two different mechanisms or genetic mechanisms of evolution. So the available toxins is going to basically be a function of the gene content, what genes you have, and again, the sequence evolution. So what the sequence of those toxins is. And then the amount that the toxin is expressed is basically regulatory evolution. So I guess I want to give everyone a couple more minutes to write that out in case they need it. So let's break these down, and we're going to look at each of these mechanisms. And today we're going to look at two of these mechanisms specifically, and then we'll talk about the third on Monday. So the first one is gene duplication. And we're going to go just through a quick example. So that you can see what I'm talking about when I say gene duplication, gene loss, changes in gene content. So here's a case that is actually characteristic of most things, for instance, within a species or usually even between species, which is that genes are conserved, which means that I have a gene and I have a copy of it. And then most other things that are in the same species as me probably have that exact same gene. And if it's functionally important, they're actually probably very similar, if not identical. So this is where the gene is completely conserved. Sometimes there are cases where there is a variance. So this might be one or two DNA bases that have changed. So these are minor changes. These are pretty typical and actually normally what we are thinking about evolution consisting of. In many cases, this is actually what we look for, small changes. A much more dynamic and frankly, in many cases, rare form of variation, though, occurs when you have a gene duplication. So if you've taken genetics, you might be aware of this. If you haven't, this is something that can occur basically a number of ways. There are genetic elements that can help create duplicates. Duplication can occur on things like crossing over. But basically, no matter how it happens, the ultimate outcome is that you have a single gene that gets duplicated. And so there are now two copies of it. And this has now created what's called copy number variation or variation in gene content. And so sometimes this happens and nothing happens with it. That individual dies off and that extra gene dies. But sometimes it actually can spread, and you can see it within multiple species, or sorry, within multiple individuals or the entire population of a species. And once we get here, we have this interesting thing because different things can happen. So one possibility is that both genes are maintained. And this would be kind of what we see, again, like very typically. But another possibility is that losses can occur. And so basically, this would mean that a duplication happens somewhere in history, and then at least one loss has occurred somewhere. So for instance, in this case, it could be that we had one duplication in this individual two, and then the original gene copy was lost. Or maybe, sorry, I went the wrong way. Maybe actually there were two losses. Maybe ancestrally there were two copies of the gene, and one was lost in one individual or lineage, and the other was lost in the other individual or lineage. So in any case, these create these scenarios where there are actually whole differences in the genes that one individual or another individual possesses that one species or another species possesses. So we're talking about this as this very like, kind of ephemeral idea. How does this actually affect venom evolution? Well, it turns out that this mechanism of variation is critically important to a group we've already looked at. So these are our Mojave rattlesnakes. And remember that they have a neurotoxic or hemotoxic type venom in most cases. And it turns out that at least for the neurotoxin, is what I'm going to use as the example, is encoded for specifically by two very particular genes. And they both have to be there and they both work together. And so the type A venoms, which are a neurotoxin, have both of these genes. Turns out that the type B venoms do not have these genes. I'm going to put this slide back up. So if you're like writing, don't worry. But I want to show you what this actually looks like. So what you're looking at here is an image. This is a graphical representation of a chunk of the genome, of a stretch of the genome. Each of these little arrows represents a gene. And the colored arrows are our toxins. And so we can actually see lots of places that both toxins are present in these two different kinds of, in this case, it might be two different snakes. But we have this very special region of the genome that occurs right here, where basically we've had something happen in the history that there are two variants of what toxin genes, because it is actually multiple, are present or can be present. And so in our neurotoxic type A species, we actually have these two genes that I've colored red that work together that are the neurotoxin that makes these snakes so potent. And then in our other species, we actually have a completely different pair of genes, and those have very different functions. And so this indicates that, in fact, gene content, just what genes you have, is a critically important axis of variation in venom. And that's really unique. It's not something we see in a lot of systems, especially not within species variation. It's not a very common mechanism of evolution, or at least I don't believe it's not widely, not what we typically think of. Probably a better way to put that. Yes, Ethan.
So then in the AB
Uh-huh.
individuals, do we see a mix of both genes together, or some of them missing, like half of this one, half of that one type of stuff?
I nodded my head, but I actually can't comment on that because the truth is we don't, to my knowledge, we don't have a genome sequence of an individual that is an AB. So those sequences came from an individual that was a type A and an individual that was type B. And I don't think anyone has published a type AB animal genome yet.
Additionally, do we see variation in the genes of A's in the north versus, like in the very northern in California versus A's in southern Mexico, or are they the same? We find the same genes throughout the
I believe, I don't know for certain. I believe that they are conserved, but there's no reason that they couldn't hypothetically vary. I mean, we know that at least that one region definitely varies and it seems to change. But I think that in most cases they're conserved. All right, so that's one axis of variation. That's really neat. It's a really cool thing. I think it underscores a really important mechanism of how evolution can operate. Yes, Sydney.
If you're gonna answer this, then obviously, but I guess you're talking about how
Yeah.
So if that's the case, then why did Venom evolve? Because like it's not necessarily like, I guess, essential for the organism to do what it does, right? It can still bite and kill organisms. It doesn't need
Sure.
Venom to do that.
Yep.
So like, what was the selective pressure that caused that to happen?
So don't think of it so much as, does it have to? I mean, that can be a case, but think about it like this. If you have a rattlesnake with, I mean, maybe not a rattlesnake, let's pick another snake that doesn't have a rattle. But if you had one that does have venom and does not have venom, which do you think will be better able to survive and reproduce?
Probably the venomous one, but like.
Yeah.
What made that happen?
We don't really know. I mean, at the moment would be what I would say. I mean, we believe that diet was ultimately the selective pressure, that if you have venom, you're better able to acquire your, your prey, but we don't have great data, to say, that's it. Okay, so I want to, yes, go ahead, McKenna.
I was just gonna make a little comment. One thing as well is that like there are certain prey species that like if a snake does have venom, they're less likely to fight back obviously because it's
Yeah.
intoxicated, whereas there are other cases where the snake doesn't get the right hold on it, prey individuals can fight back and actually cause like significant damage to the snake. So that could have been like depending on what the prey is that we're talking about, that could have been the pressure that contributed to the evolution of that. Right, and it can start with a very small variation because they're modified salivary.
Yeah.
So whatever has that initial little bit of toxin would obviously be just a little bit more effective. So that could have been like a little like kickstart for it.
Yeah. We'll also talk about actually another kind of way that evolution occurs that is a little different on Monday. So
Do we have any like common ancestors of snakes that display it in them? That would be like transitionary species that would display them as like where they got it from, I guess.
So I guess, I mean, in some ways, the closest that we have is what we assume is representative of some of the common ancestors would be things like epistoglyph snakes. So, I mean, and obviously in those cases, like I was saying, we have cases where sometimes they need venom and sometimes they don't. Sometimes it's a critical part of their life history. Sometimes it might not be. So there kind of seems to be a spectrum. Yeah. Okay. So the other mechanism that I want to talk about today is another one that's really easy to think about. This is gene regulation. So I ignore that text that says gene duplications and loss. What I want to talk about here is how expression can differ and create different phenotypes. So you can imagine a scenario where you have two genes. This is sort of the same setup as the other, except instead of looking at gene content, we're going to look at expression. So in this scenario, we have two individuals, the same gene, and they're expressing them equally. If we see this and if the genes are conserved, we actually expect that these should lead to identical phenotypes. If instead, however, we see variation in expression, differences in expression, here we've got a case where gene 1 is perhaps transcribing a lot more of that gene. And in individual 2, we're transcribing much less. These are going to be very different phenotypes. These are going to be cases where you might have a lot of one protein produced in one individual or species, and very little in another individual or species. And in fact, we have a really cool case where this is true as well. And guess what? You've already seen it. So this is going back to our palm pit vipers. And one of the things that's kind of interesting here, and I didn't actually drop this when I talked about them the other day. In fact, I mentioned that this guy has a very potent neurotoxin that was characteristic of that lineage. Turns out that neurotoxin is homologous to what we see in rattlesnakes, which is really cool, really interesting. But not something we're going to talk about too much, because what I really want to actually draw a contrast between is that we just saw an example where we have a rattlesnake with this potent neurotoxin, and the variation in the venom phenotype is mediated by the gene content. Here, it is actually mediated by expression. So if we look at the genome, or sorry, the gene expression of this other individual, which we know has a very different venom phenotype, we actually see that these two toxins, which are the neurotoxin in our black speckled palm pit viper, are still present and still being very lowly expressed in this other species, our Talamancan palm pit viper is its common name. So this is kind of cool because it's, in a way, it's convergent evolution of this neurotoxic hemotoxic dichotomy, but it's occurring through a very different genetic mechanism. Yeah. And so this is really important, obviously, because we know we now have evidence, or this is just an example, really, of variation in venom occurring through changes in gene expression. And we kind of already knew this. This is actually a really dramatic example, but there are lots of cases where we see differences, for instance, in populations in their venom that occur primarily through gene expression. And one of the reasons that we expect this is that gene expression can evolve very rapidly. We expect gene duplication events to be kind of a rare thing. And in contrast, there are lots of different genetic mechanisms that can change gene expression. And in fact, it could be that early changes in venom composition are primarily a function of regulatory evolution. So for instance, when you have early divergence of a lineage or separation of populations, it may be that the way that these lineages are fine-tuning their venoms to their environment is primarily through changes in regulation of the venom genes. All right, so that was a bunch of very genetic and evolution kind of heavy stuff. So I thought this would be a great time to take our stretch break. So 5 minutes. And once you guys get back, we're going to talk about one more really cool instance of venom evolution. And I'm going to make sure that, yep, checking that. Okay, so what I want to talk about now, and the last thing I'm going to talk about lecture-wise, is actually the origin of venom in reptiles specifically. And if you're talking about the origin of venom in reptiles, at least in snakes, you basically can't mention it without talking about this really seminal work that was done in 2006, I think, looking at the evolution, both of these lineages and of some other things. So in 2006, there was this really important study that did a molecular phylogeny of some lizard families and snakes. And it identified this clade that I think that you all have already talked about called the toxicofera. And that includes all our snakes, as well as these families, the Helodromatidae, the Anguidae, the Varanidae, Iguanidae. And so it's really important to note that that is a really key part of this, that there's this monophyly of this group. And I also want to say that when that came out, there was, some contention. Some said, no, these are not actually monophyletic lineages, that the data are not well representing the phylogeny. But more recently, there have been genomic studies, so leveraging huge amounts of data, and this relationship has been supported. So to the best of my knowledge, there's every reason to believe that the Toxicofera exists as a clade. And in this same paper, one of the things that they noted was that this family, you want me to go back? Yep. So I'll start talking about the things on the next slide while we're still copying some notes. So in this same study, what they looked at were a few different things. They looked at the morphology of these organisms, and they were specifically interested in the oral morphology. And they also did some they used some genomic techniques to sequence the proteins, or the DNA of the proteins being produced in these glands. So they identified, again, in many of these lizards, these oral glands. One of the things to note is that unlike snakes, these glands are, for the most part, on the lower jaw, but that's consistent with the Helodromatidae. I'm going to add some text up here. So in some cases, these glands, the DNA that they got out of it, were toxin genes that were being expressed. or at least they seem like toxin genes. And so because of this, led the authors concluded and put forward this hypothesis that in fact, venom, broadly speaking, evolved quite early or earlier than we had thought of in the reptile system. And so it would argue that in fact, the Helodromatidae, the common ancestor of Helodromatidae and snakes, was venomous in some capacity. Yes, Ethan.
Do you know if, like, say we want to look at the fossil record at some point,
Mhm.
for example, available,
Yep.
if we wanted to, is there any way to look at the fossil origins, and is there any sort of like facial structures that kind of point to venom possibilities?
I don't have the expertise to answer that. I would say, I mean, there are certain hallmarks we could certainly try to look for. Things like grooved teeth that look like they would facilitate venom delivery. You might also look for some kind of jaw morphology that looks like there's an associated gland, but I don't know how possible those sorts of things are to identify, to be honest. So let's see what else we have here. I don't know why it's not moving. There we go. So there's this big key implication that came out of this study. Again, that venom is a derived trait in the toxicoferan clade. And thus all species in the toxicofera, from an evolutionary standpoint, are venomous. And so there are some examples of places that are probably like, wow, that makes a lot of sense. So not too long after this study came out, just a few years, there was another study that implicated, or it was actually specifically designed to look at these guys, these are Komodo dragons, and implicated venom as being a mechanism through which they're getting their prey. So they basically said varanid lizards are venomous, or Komodo dragons are venomous. Out of curiosity, has anyone heard this or read it or seen anything about it? Okay, Ethan. Yeah, so a lot of people. So you guys are familiar with this idea. Komodo dragons are venomous, right? Cool, neat, fine, awesome. How about bearded dragons? Does anyone have a bearded dragon? No, okay, well, I do. My wife does. So, but the thing is, and this is not like really an exaggeration, because a bearded dragon was one of the taxa used in that previous study. And so it was basically implied that the bearded dragon was venomous at some level. So you might think, wait, what? Like, that's interesting. It's kind of an odd conclusion. Maybe, maybe not. So in some cases, people basically said, I don't believe that. I think that this is a very bold claim. And it became a question of, let's test this idea. And there have been a couple of studies that came out. There were actually immediately after this publication, there were people that just wrote frank responses and said, this is why this is a bad idea to use this kind of language. And they dealt with very logistical things like, it implies that bearded dragons are venomous. And so does that mean that every bearded dragon owner now needs to say that their pet is venomous? Because that means a lot of things to a lot of people. What if I have a python? Do I need to now classify my python as venomous? And what if my state has laws against owning venomous pets? So in some cases, they're being a little hyperbolic, but it's sort of highlighting this intersection of philosophical and logistical issues that can arise with this kind of classification. But there were also some scientific critiques. So one of the problems that occurred, or one of the critiques that people have mentioned, is that in that study where they said, these are toxins that we're seeing in the oral gland, the way that they defined toxin was by homology. So we've talked now a few times about homologous structures. That's basically when you have the same ancestor. It turns out that genes can be homologous as well. So if you think about the fact that, if I have a gene that was ancestral, or if my common ancestor has a gene that I have and a mouse has, those genes are homologous. And that can be true for a lot of different, or basically every gene. And so in this study, they were defining genes based on homology. They made a phylogeny of the genes and they said, hey, this is a snake toxin and this is another snake toxin. And this thing that I found in the lizard venom gland or the lizard oral gland, it is in sort of this same clade. But the problem is that homology does not always define function. So there are lots of cases where functions change even among homologous relationships. We could think of some really obvious examples. So for instance, your arm is homologous to a bird wing or a bat wing, but they could have very different functions. And so it could be that even though that these lizards have these homologous genes, these homologous proteins, they're not necessarily using them for prey capture like we see in venomous snakes. And then there were some other studies that said, well, here's another thing. A lot of these homologous toxins that you're identifying, if you look at different body tissues, they're expressed all over the place. So for instance, if it's really like a potent toxin that we see in a snake venom gland, we would really probably expect that it's not going to be a lot of other places. There's only so many body tissues that are evolutionarily sort of adapted to produce and store those toxins. And so if you're producing some of these things in tissues like your muscle tissue or your liver or your stomach or your brain even, it argues that maybe these are not in fact the toxins that we're thinking that they are. And as a consequence, this is kind of arguing that they don't have the specialized function. Maybe they're not actually venomous. And that's actually kind of where we are these days, because there's a lot of things that we still don't know. And we're basically faced with a condition where in a lot of cases, the function of these different genes is in doubt. We don't know. And there tends to be kind of two sides to this argument. People that are a big proponent of the toxicofera venomous theory or hypothesis basically say, we don't know what these genes do in some cases, but we know that they are a conserved evolutionary trait because, again, they arose in this ancestor. We know that they're important, or we believe that they're important. And we should realize that our human definition of venomous does not correspond with maybe what evolution, what it meant in an evolutionary sense. And so really we should consider all of these things as venomous and realize that maybe our human definition of it is just not consistent with the actual historical evolution of the trait. But then there's another argument, and I actually coded these as argument 1 and argument A because I didn't want to give either a sort of advantage as 1 or 2. And this argument says, we don't know what these things do, so we can't call them venomous because our definition of venomous intrinsically implies a function, a specific function. And so the more conservative approach is actually to consider these things as non-venomous, at least until we know what is actually going on, until we have stronger evidence that in some cases they're actually serving a function in digestion or dietary acquisition. And so that's kind of where we're at. And I think that I've always thought this is really interesting. I'm happy that I personally don't have a particularly strong horse in the race, like there are other scientists that do, but I'm really interested to hear what other people have to say about it. And so this is something I'd like you all to think about. Yes, well, yeah, absolutely. So what we're going to do for the rest of this class period is I'd actually like for you all to debate this topic amongst yourselves. So you're already kind of split into groups because we're just going to use these tables. And Dr. Gangloff and Princeton and I will each kind of come to a table and we'll be kind of mediating a debate. We'd like for you all, ideally, there are going to be some differing opinions at each table to talk about this idea. And really what I want you to address is to take sides on this issue. Should we consider all organisms in the toxicofera venomous? If it helps you think about it, maybe think about the bearded dragon and the Komodo dragon. Should we consider both of these things venomous, or is only one venomous? And so after we've talked about that for a little bit, we have, I put a little QR code up here. You'll be able to do a quick reflection on it, just thinking about that topic. And I want to get everyone's opinion on what you really, truly think after you've heard all the arguments. And then that will be it. So any questions before we get started? All right. Let's break up. You guys have a preference of what table you would like to do? Dr. Gangloff, you would
I feel like I'm going more with I don't know, like especially when you
So just for some clarification, if you guys go to this form, you can see I've got a couple of prompt questions. They say basically, what do you think is the best argument in favor? What do you think is the best argument against? And what are your thoughts? I would use those first two as your jumping off points. So trying, as these big groups, what do you think are the best pro and con points? And then make sure that when you're doing the last question, what is your opinion? That's sort of your own thoughts. And then make sure to get that submitted before you take off. Cool.
to kind of work through it, discuss it, and then write your own answer.
All right, you guys, where are we at over here? Argument A.
There was a request to go back to the slide.
Oh, yeah, absolutely.
I don't think we should necessarily get stuck at the bottom, so.
That's what I'm trying to write right
I'll go back to that other slide.
Like, for example, on this thing, that's at the zoo, those will be labeled as venomous. But to us, they, when they have significant danger, usually we're venomous use. Yeah.
But then we also talked about.
Can I just make an argument for me?
Someone's argument three.
You should just face it up.
So then that would be argument B. That would be argument A. That right now,
Other than like obviously
wouldn't
right
know.
now,
It goes as
tiger.
potentially injurious.
The way it's
So
worded, I
if
still
it's been
disagree though.
potentially harmed
You know what
human.
I mean? So, like-
oh, you're right. It is actually worded for it.
Exactly. So there's a lot of.
The idea is that you would take it on a need by need basis. So the argument basically of A is not that everything.
What I would personally
It's more that we don't, that nothing should be considered venomous until we know what it is.
Then A. It's perfectly what I wanted. I was like a non-venomous person. I was like, well, that's kind of dangerous to assume until you figure it out.
Yeah. The idea is like, if you know it's venomous, but you're saying it's venomous. But for instance, the bearded dragon, they would say it's non-venomous.
everyone in the class has the ability to migrate. All of us on enough to so it's really all
The function of it.
It has this- What if those situations are probably pathogen diagnosis?
Now I'm gonna dramatically introduce all of the functions.
Right? So that's sort of their point is that we don't actually get to
water tank, for example, and they have an anticoagulation in their saliva, which you can consider about
sort of in the same way it
prays, but it's not gonna like, all it does is it's just. So from the standpoint that it is benefits, but nobody really goes like on the benefits.
Exactly. There's no reason to be afraid. What if you sort of say, oh, it's venomous. They're not gonna figure out what's going Yeah, I know. Wait, so we need to figure out pro of
So you don't think it's got an evolutionary context?
Yeah, theoretically, somebody
Yeah.
See, I would say personally, it's not all
Yeah.
The first question is, what do you think is the greatest evidence for
I mean, theoretically, if you had a bearded dragon, if something happened, genetically like crazy, so it might just come out. I don't know.
So you don't
Like
think that.
in the sense of like probably. Anybody agree?
Yeah. That's a fair.
Anybody has any sort of idea why someone would?
Why someone would what?
Agree with our. Oh, I like that. That makes sense.
How are table design? No, I'll give you an example. So, argument two. So
that's why
the logic behind argument two, this is at least for components of argument one. The logic behind argument two would say that whales.
Yeah, they're not including like.
Because the idea behind argument A or argument two is it's based on function, from a functional standpoint,
they're not.
a whale is in the water, is not lifted, it's eating fish, it's never getting out. That is a fish, it's doing everything a fish does.
I mean, I would say two.
But if you look at it in an evolutionary context, yeah. And so that's the argument here. In evolution, they're saying
But is it not
definitely
like, could be, have something that's alive.
evolutionarily.
That slows down the heart, or slow move in some way.
Like it.
Same thing I do with starter
Makes
things.
sense, though, because like when you describe a whale, like that's big.
But isn't fish, when you think of a whale,
So-
you shouldn't be.
It's not like legitimately a fish, but it.
But you just said it's not
So-
legitimately a fish. So that means you just said it's not legitimately a fish. So that means legitimately a bearded dragon is venomous.
I'm saying, but when you
Under that.
describe it like that, under that argument, it is, so like it's not necessarily a wrong argument.
So is that right or wrong though?
Are you think a whale should be a fish?
What's another way to?
It's crazy. It's funny because like this really is. But there was really, that is the hardest thing. They would say, how could you say that they're not venomous?
What is your get?
No, it's a totally different concept. It's not. It's the exact same concept.
So they're not venomous.
I mean, it is gay, so it's
I'm so stuck on the whole.
Because it's creating.
Because like, you can't just assume everything that's related to what you are saying.
Itself, what's sitting in this platform is.
So they're not venomous.
The components of each side actually both think they both think they know exactly what it is. They both think they know.
Look at that.
The hard components of either side, they can
We look behind something and then.
I need more parts.
As a breathing. Something that helps the light.
Yeah. I mean, it makes sense. If you can have genes that make the same aren't expressed, just because you have a gene or something doesn't mean that you're
Well, yeah, but you could also.
expressing it. And then you're using that protein in the same way, but that event is the same way. I guess that's pretty cool. But it's kind of intended function.
That makes sense.